I will talk today about our most recent findings on the meningeal immunity, and it's very nice because I noticed here that it's a very diverse public, so it's very important to share all this clinical with our experimental models. It's very important to push forward the system, and as Judy mentioned before, so I'll talk about meningeal gamma delta T cells, and I'll be focused on this molecule called L17, which is nowadays is very popular when we talk about autism models. So as an outline, I would just briefly give a brief introduction. So we work very closely on the relationship between the immune and the central nervous system. And we really think that these both systems, they co-evolve together, and several key molecules in both sides, they are just cross-talking every time. And we are starting to appreciate a lot this neuroimmune communication. So then why and how anti-pathogen molecules may have co-evolved with host behavior. And I'll talk a little bit about the maternal models of autism, and a lot of literature of IL-17 driving these abnormal social behaviors. So then I will spend most of the time talking about how IL-17 coming from the meningeal spaces regulates anxiety-like behavior under homeostasis. Unfortunately, we didn't investigate a lot under disease condition. This is our next step, but I would just show briefly here. So how we think the cytokine is also important to regulate anxiety-like behavior. It's important to mention that anxiety, I guess, is around 40 to 50% of autistic kids. They show anxiety behavior as well. So it's quite important, and then you can have a quite interesting connection here between anxiety and autism. And by the end, I hope we can all appreciate the potential involvement of the meningeal-derived L17 in neuropsychiatric diseases, and this is our next step that we are focusing right now. So as I said, so in our lab, we're very interested in this neuroimmune connection, and we really think these both systems, they co-evolved. So probably throughout the evolution of the vertebrates, and over here I just highlighting the human evolution, it's likely that the individuals, they were selected based on their capability to efficiently fight against pathogens, because we had a very highly infective environment back then. So at the same time, it was likely very important to develop a very efficient immune system to promote tissue repair. And in order to increase our chances to survival, several behavioral traits, they were also selected. And for example, just highlighting here in this cartoon, alarm and avoidance, in order to avoid unnecessary exposure to these pathogens, rivals, and predators and the environment, to make sure that you will survive. And among these behavioral traits, we can highlight here the selection of physiological levels of anxiety as a default mode, because it's important to be aware of your environment to avoid these unnecessary risks. And what we are learning nowadays is that the immune and the central nervous system, they share common signaling pathways, and that they are more connected than we expected years before. So infections and some specific bacterial communities have been correlated with psychiatric diseases. And we are starting to appreciate right now a lot about anxiety, depression, and autism, and these commensals. And social stress have also showed huge impact on the immune system. So they're really connected. So maybe we have cytokines which were selected over time to do two things at the same time: fight against pathogens, but at the same time shape behavior. And the other side is true. So neurotransmitters, they are not only regulating behavior, but they are also regulating the immune system. As a very good example, and a very classical is of this neuroimmune interaction, we highlight the series of very elegant studies published a few years ago by Glorious Choi, Junhu, and then Lichman's lab, which shows that the prenatal exposure of viral components, like for example here showed by poly-IC injection in pregnant mom, it's linked to social behavioral deficits in experimental models of autism. More specifically, so these viral components, they are generating this unique population of Th17 cells in the gut, together with commensal bacteria, highlighted here by segmented filamentous bacteria, which we just named SFB. And specifically, they drive these behavioral abnormalities, specifically in this cortical region called S1DZ. And then that's why we see in the offspring all these behavioral deficits. It's very important because a lot of literature have linked infections and viral infections during pregnancy with autism-like behavior. So early this year in January, the complexity of this neuroimmune interaction was appreciated again by the demonstration that fever episodes and the consequent improvement of autistic behavior was related again with IL-17. So in this case here, I'm just highlighting that if you just give a shot of LPS in mice in these MIA models, so you have induction of fever, and we know that fever it's important to improve the autistic behavior. And then we can see basically here in this MIA model when we inject LPS, so now they get social again. And then it was very interesting because IL-17, what was bad during the pregnancy, during the prenatal phases, right now it was important to recover, to rescue this social behavior. So for example, if you inject anti-IL-17 together with LPS, so we don't have this improvement in the social behavior anymore. Suggesting that IL-17 signaling is very complex and is really regulating, depending on the phase, the host behavior. However, the focus of my talk today is regarding the role of the same cytokine, IL-17, under homeostasis. And the main focus of our lab is particular these meningeal layers, which basically is these 3-layer membranes which are covering and protecting the brain. And we are trying to suggest that not only the gut, but the meningeal spaces are also a source, a very, very strong source of this unique cytokine, IL-17. Given the physical proximity between the meninges and the brain, the meninges are very attractive to target future therapies aiming the treatment of neurological disease. And now we know that these spaces, they harbor a very rich and diverse immune system, even under homeostasis. So over here, highlighting so how rich in terms of cell diversity the duromeninges are under homeostasis. And we can basically find every single immune cell over there, B cells, NKT, gamma delta T cells, which is the focus of my talk today, CD4 and CD8 cells. The idea here is that since they are very close, cytokines and other soluble factors are released by the duromeninges and they are reaching the brain, and then we can have the proper neuronal function. So a few years ago, our lab has published that the meningeal-derived IL-7 interferon gamma coming from T cells in the meningeal spaces regulates also social behavior. So basically, when you have interferon gamma and you have these T cells in the dura, so you have the proper circuitry in the prefrontal cortex. And mice lacking on interferon gamma or CD4 cells in the dural spaces, they had issues in terms of sociability, suggesting that these meningeal spaces are also important in driving social behavior, but in this case, under homeostasis. In our work, so we have like this very basic and simple triad here that the immune system, pathogens, and also commensals, they are all connected with host behavior. So we can, if you have changes in your microbiome, or if you have any certain infections, so you can shape your immune system and you can shape host behavior. So this is everything in a very, very well controlled under homeostasis. But if something goes wrong with the either immune system or with your microbiome, so you can have issues, and then we can have the development of neurological diseases. So we, as I said, we are very interested in the T cell populations and the dural spaces. And this is just a regular mass cytometry, and you can appreciate here how diverse in terms of immune composition the steady state meninges are. So most of these populations here are macrophages, but we also find B cells, we also find neutrophils. And when we start this study that I will show today, we are looking for these T cells to try to investigate like other factors that these T cells were maybe releasing. And I noticed by like that time that there was a very tiny but very consistent population of double negative T cells. What I mean by double negative, they were CD3, so which is a marker of T cell, but they're not expressing CD4 or CD8. By that time, what we did, we basically stained for their TCR, and then we found that 50% of these double negative cells were expressing the TCR gamma delta. TCR gamma delta are a very, very innate like T cell in terms of evolution. They are the most ancient T cell appearing in the immune system. And they are harboring the barrier spaces. So we find a lot of gamma delta T cells in skin, in the gut, in lungs. But we were very surprised by that time that we found a lot of gamma delta T cells in the dura. Here is just to have a spatial location of this cell. So these are the big blood vessels that we can find in the dural spaces, which are called sinuses. And most of these cells, they were just sitting along the sinuses in the meninges parenchyma under homeostasis. So as we're interested in these neuroimmune connections, and we are mainly focused on cytokines, we next investigate what kind of cytokines these gamma delta T cells were making. And as you guys can see here, the alpha beta T cells, which are the conventional T cells, they are most towards interferon gamma production. But you are very surprised by that time that over 50% of the gamma delta T cells in the dural spaces, they were making IL-17. So when we checked about any other closest population of the brain making IL-17, we could quantify that the gamma delta T cells in the dural spaces, they were responsible for over 95% of all IL-17 under homeostasis. So which was very surprising. So the other way that we could characterize deeper these gamma delta T cells was based on their migration kinetics to the dural spaces. We also saw that most of these gamma delta T cells were expressing this V gamma 4 TCR, which is shared for gamma delta T cells in other barrier tissues, like for example, uterus, lung, and tongue. And it's very interesting here because these V gamma 4 gamma delta T cells, they are just generated during the prenatal stages. They are not generated after birth. So which was very surprised as well. When we started to collect the dura from different time points, we found that the gamma delta T cells, they were not there in the prenatal stages, even though they were generated during embryonic phases. But as soon as P3, so we start to find that substantial numbers of these cells, they're already seeding the dural spaces. And at P7, almost the same amount as the number of gamma delta T cells in adulthood. So we also stained for Ki67 to evaluate how much proliferation how much these cells are proliferating. And we found that at P1, P3, and P7, so these cells, they were very, very highly active, proliferating in the meningeal spaces. And nothing really was happening during adulthood, suggesting that after birth, as soon as the mouse is born, so these cells, they start to migrate to the dura, and then they are actively proliferating and to fill the space. We also did single cell RNA sequencing in order to define and to have a better understanding in terms of the genes expressed by these cells. And we basically isolated gamma delta T cells from P7 and also from adults. And we compared these cells with gamma delta T cells isolated from spleen. As you can see here, they are very different compared with splenic gamma delta T cells. And we could take this data set to see what kind of chemokine receptor were expressed on these populations, to try to identify what kind of signals were driving their migration to the dura. We found that the chemokine receptor CXCR6 was highly expressed in these meningeal gamma delta T cells, regardless the age, P7 and adults. So then we just try to validate this data coming from the single cell, and because it was just mRNA expression. And we took advantage of this CXCR6 GFP mice, and we could see that indeed gamma delta T cells in the meningeal spaces, they were highly expressing CXCR6. So then the next, what we did, we just basically took it out many gamma delta T cells from CXCR6 deficient or CXCR6 sufficient mice. And we could appreciate that in the absence of CXCR6, we saw lower frequency and absolute numbers of gamma delta T cells in the dura. So then together, this data suggested that the gamma delta T cells, they were arriving very early, postnatally in the dura, and probably CXCR6, the chemokine receptor, was very important for the recruitment or retention in the meningeal spaces. As a summary for this first part, so we saw here that the meninges, they harbor this tissue resident population of innate-like gamma delta T cells. They are probably the nearest CNS source of IL-17 under homeostasis. So we also found that the gamma delta T cells are likely generated during prenatal stages, and they seed meninges shortly after birth. For the last, we found that CXCR6 contributes to gamma delta T cell recruitment and or retention in the meningeal spaces. As our lab is very interested in these neuroimmune interactions, and we are very interested to know how cytokines and the immune composition is shaping host behavior, we next address, so what's changing if we could see any differences in terms of behavior in mice lacking gamma delta T cells. So we basically use this gamma delta T cell knockouts, and we did a battery of behavioral experiments. We didn't find any difference comparing social or learning behavior, suggesting that these populations of gamma delta T cells are not really involved in sociability and learning under homeostasis. But we found a very striking and very interesting differences when we did behavioral assessing anxiety. So the way that we assess anxiety in rodents, which is validated by all the anxiolytic and antidepressant drugs nowadays in the market, is by using these two paradigms. They are named elevated plasmase and open field. So basically, these are very simple paradigms, and they are based on the approach avoidance behavior of mice. So for example, if you place a rodent, a mouse here, in the center of this maze, so most of the time they will spend in these closed arms, because just by nature, they feel protected in the darker and in the closed arms when compared to the open areas. At the same time, they have this conflict because they also want to explore new environments. So then you can measure how much time they spend in the closed versus the open arms. Usually most of the time they spend on the closed. The same is true for the open field. So the open field is a very simple paradigm, is a box, and then you also just place the mouse in the center of this box, and you assess how much time they will spend in the corner or in the center. As they spend more time in the center of the open field or in the open arms, you measure this as anxiety or not. But right here, we are just doing under homeostasis. What we found by that time is that animals lacking on gamma delta T cells, they were spending more time in the open arms compared with their wild type litter mates. And the same was true for the open field. They were also spending more time in the center of the box. So then suggesting that under homeostasis, gamma delta T cells, they're probably keeping these guys more vigilant, more aware, or probably we can interpret here that they were less anxious at baseline compared with wild type mice. But of course, these we are just using total knockout, so TCR gamma delta full knockout, and gamma delta T cells, as I said before, they are residing in other tissue barriers. Another thing by doing this experiment only with the knockouts is that we could really isolate the actual effect of these cells, because the gamma delta T cells knockout, they don't have gamma delta T cells never during development. So in our lab, we have this quite specific way to target the dural spaces and also the brain by doing injections into the CSF, the cerebral spinal fluid. So basically, if you inject the antibody or cytokines by doing ICM injections, we can target the brain or we can target the neural spaces. Because as you can see here in this cartoon, if I inject an antibody here, it's going to end up or in the brain or in the neural spaces. So next, we try to target better the meningeal spaces in order to isolate the peripheral effects of gamma delta T cells in our paradigms. So, as I expected, and also pointing out the important role of gamma delta T cells in the dura, so we saw that 3 days after injection of anti-TCR gamma delta, so these mice, they were also spending more time in the open arms compared with the isotype control. And the same was true also for the open field paradigm. They were spending now more time in the center of the cage. So this data was very interesting. It was very important to conclude that the effect that we saw was not a specific, like something that was happening during development, and also probably was something more specific to the meningeal spaces, highlighting the importance of these neural spaces of controlling the normal behavior. So after this, we tried to identify which are the mechanisms downstream of these gamma delta T cells, what they are making, what's happening. So we basically repeated the same injections with the anti-TCR gamma delta, and after 3 days, the same time point that we use for the behavioral assays, we tried to identify what was happening with their TCR and what was happening with IL-17. As you can appreciate here, when injecting anti-TCR gamma delta, we found that TCR internalization that was happening 3 days after the injection. These cells, they're actually not dying, they're not eliminated, they're not deleted from the system. They are just internalizing their TCR. So as they are internalizing their TCR, we hypothesize that maybe, okay, they don't have more TCR in their surface, so now they cannot be activated properly. What about their IL-17 production? And we basically just confirmed this hypothesis that when we gave anti-TCR gamma delta, so these cells are now making less IL-17 compared with the isotype control, suggesting that the TCR activation, it's important to keep the cells releasing IL-17 under homeostasis. And this is probably the mechanism by which they are controlling anxiety behavior. So then for this part 2, what we saw is that the meningeal gamma delta T cell, they regulate the host vigilance state, which are naming here anxiety-like behavior. And the TCR engagement contributes to the IL-17 production by these cells. So it's something that is, it's happening constantly. It's also important to mention here that we did all this experiment 3 days after the antibody injection. When you repeated the same behavioral paradigms after 7 days, so everything was back to the normal, suggesting that is a reciprocal control of IL-17 and anxiety-like behavior. The downside about these previous experiments that I was showing is that every time I was measuring IL-17, we were doing ex vivo stimulation. So then it's not really showing what's happening in vivo, and if these cells are really secreting IL-17 in vivo. So then we took advantage of this IL-17 GFP reporter. So these guys, every time you have IL-17 transcription, so the cells, they will become green. So then you don't have to do any ex vivo manipulation, and you can measure the IL-17 activity in vivo. It's a more reliable readout of IL-17. As you can see here, when you isolate gamma delta T cells from P7 or adult mice, so they were already transcribing a lot of IL-17. And this is very striking for P7 when they just get into the dura. Like over 50% of the cells, they were actually actively transcribing IL-17 in vivo, suggesting that IL-17 is really active. These meningeal gamma delta T cells are really active in vivo. Nothing really what's happening with the conventional alpha beta T cells, suggesting that these guys are the main players in regulating anxiety-like behavior. So we also saw that the gamma delta T cells in meninges, they were very active compared with gamma delta T cells in spleen. And we could also validate this IL-17 production when you compare the IL-17 measurement in wild type meninges versus TCR gamma delta knockouts, where the measurement was gone, suggesting that they're really releasing and they're the main source of IL-17 in the neural spaces.
Hi, Kalil. I'm sorry, I don't want to interrupt, but we are running. You've got 3 minutes which includes questions. So
okay, yeah, okay. So I will just speed up here. So then the next, we just tried to find what was driving IL-17 production in these cells. Then we found very interesting this correlation between commensals and IL-17 production. When we treated these mice with antibiotics, we found that this production of IL-17 was downregulating, suggesting that commensals are driving IL-17. We did the opposite experiment. We basically inject LPS in periphery, and we found that they are also upregulating IL-17, suggesting that these cells, they sense peripheral and commensal signals. So then, just to speed up here, so we found like the same was true when we blocked IL-17 into the CSF. So basically, the mice, they recapitulated the same phenotype. And when you gave IL-17 back to the TCR gamma delta knockout mice, they also increased their anxiety levels, suggesting that IL-17 was the main player. So just to finish here, so what kind of cells were responding for IL-17? We found that the prefrontal cortex, it's a really important brain region involved in anxiety. The neurons on these neurons, the neurons on this region, they were expressing high levels of IL-17 receptor, specifically here excitatory neurons. We also did single cell RNA sequencing on this neuron, and we could find several genes differently expressed when you took it out IL-17 from these neurons. And it's very interesting to notice here that most of these genes, they're involved with anxiety and depression in humans. And most of the genes which are differently expressed, they were involved with GABAergic synapses, which is a main target for anxiolytic drugs. By the end, so by taking out the IL-17 receptor from neurons, we could recapitulate this phenotype, showing that the connection between gamma delta T cells and neurons in the prefrontal cortex, they are regulating anxiety levels in mice. So then as closing remarks, so we show that the meningeal arbor populations of gamma delta T cells, they are the near source of IL-17 under homeostasis. They support the host vigilance, and neurons in the prefrontal cortex, they are regulating anxiety-like behavior. So, and just to finish here, I'd like to thank you all. Sorry for running out of time. And I would like to thank my lab, so Yoni specifically, all the people which work closely with me during this study, and University of Virginia, where we developed this study, and now our new home and Washington University and so on. Thank you so much.
Thanks so much. That was an amazing presentation. And as I said, I love that area of work because it helps all of us looking at neuroimmunology. We had one question that came into the general chat that I think is that you could, if you can answer quickly, is how does IL-17 modulation interface with Dr. Hustak last week regarding IL-17 activation role for benefit as a beneficial role? If I'm not sure if you're familiar with that work. Sorry,
can you repeat it?
Sure. Yeah. Sorry, I'm reading quickly too. So last Sunday, Dr. Junho spoke on IL-17 activation and the role of IL-17 activation for beneficial, had a benefit in the MIA model. So the question, and I think it's a really interesting question, is how does IL-17 modulation that you're talking about interface with?
Oh yeah, this is it's very interesting, and we really love all these studies from John Hu lab and Gloria Choi. So it's very, the IL-17 signaling neuron is very complex, and if you go back for their same group, they show that in MIA models, IL-17 signaling is bad for drive, impairment, and social behavior if you have IL-17 signaling in the prenatal stages. But other than that, in adulthood, IL-17 is good for social behavior. It's very complicated nowadays. We don't know how to link these two phenotypes because IL-17, we saw that is expressed in several other brain regions. So I think it depends on which region IL-17 is driving signaling and which phase of the life you have IL-17 signaling, the outcome is going to be different. So it's something that we are trying to investigate and to understand better nowadays.